Welcome back everyone. Today we learn about the kidney. Before we explore the various structures of the kidney, let's have a look at the urinary system, in which the kidney is part of. The main function of the kidneys is to filter blood and to remove excess salts, water and nitrogenous waste such as urine. The ureter transports urine from the kidney to be stored at the bladder via peristalsis using its muscular walls. The bladder is a muscular elastic organ that stores urine. When the bladder wall is stretched, the nervous system is signaled, which results in urination. The sphincter muscle is a circular muscle that automatically controls the excretion of urine when the muscle relaxes. Lastly, we have the urethra, which is a tube that carries urine from the bladder. In males, the urethra also serves as the passage for sperms. Now, let's zoom into the parts of the kidney. First, we have the cortex, which is the outer dark red region. The cortex is covered and protected by a fibrous capsule. Next, we have the medulla, which is the inner pale red region. It contains renal pyramids, which are conical structures. Within the renal pyramids are numerous nephrons. The nephrons are tiny kidney tubules in which urine is formed. We also have the renal artery. The blood flows into the kidney via this artery that allows exchange of water and ions in the nephrons. Lastly, we have the renal vein. Blood that leaves the kidney has been cleansed, whereby excessive substances would be removed. We will now explore the nephron and its function. The first part of the nephron we will look at is the Bowman's capsule and the glomerulus. The main process that occurs here is ultrafiltration. The basement membrane, which is partially permeable, wraps around the glomerular blood capillaries. The basement membrane only allows water and other small molecules such as glucose, amino acids, mineral salts, and nitrogenous waste products to exit the glomerulus and enter the Bowman's capsule. Large molecules like proteins, fats, blood cells, and platelets are retained in glomerular capillaries. Do note that the diameter of the efferent arteriole is smaller than the afferent arteriole. This creates a high hydrostatic pressure, which is the main driving force for ultrafiltration. The second part of the nephron we will look at is the proximal convoluted tubule. The main processes occurring here are selective reabsorption and osmosis of water. There is reabsorption of 85% of the substances, specifically glucose, amino acids, lactic acids, water-soluble vitamins and ions into the interstitial fluids to prevent electrolyte imbalance. With it being highly convoluted, it increases the surface area to volume ratio to maximize reabsorption. The third part of the nephron we will look at is the loop of Henle. The main process that occurs here is reabsorption of salt and water. The loop of Henle is closely associated with the parallel capillary system. Blood flow is in the opposite direction as filtrate in nephron, which helps maintain a steep concentration gradient. Osmosis of water occurs at the descending limb. At the ascending limb, diffusion of salt occurs at the thin segment due to the high salt concentration in filtrate. Also at the ascending limb, active transport of salt occurs at the thick segment. The fourth part of the nephron we will look at is the distal convoluted tubule and the collecting duct. The main process occurring here is the osmosis of water. When the body is dehydrated, antidiuretic hormone, ADH, which is produced by the hypothalamus, which is then stored in the pituitary gland, is released. The membrane of the distal convoluted tubule and membrane of the collecting ducts become more permeable to water when ADH is released. That's all for today. Thank you for the effort to learn with me. Stay tuned for more interesting lessons. See you next time. Bye-bye.